Hey guys, welcome to the disease unit lesson 8 review video. I really liked how we're using Nearpod and how many people are doing that, so that's awesome. In this lesson, when you were learning about meiosis, I made some interesting models on my kitchen table, and you could hear my voice as you went through the meiosis steps, and then you just had to do your vocabulary and your work. Okay, so first, modeling meiosis. What some people say meiosis, meiosis. Just a matter of preference. So a few notes that we want to put down here. I put the Amoeba Sisters up here if you wanted to listen to them, or there's a ton of videos on YouTube if you're still having some struggles, okay? So remember, interphase 1, interphase is whenever we have our chromosomes kind of condensing, going from the squiggly DNA that it used to be up in the nucleus into individual chromosomes. Okay, so that's when our interphase is. We've got our prophase. The big guy about prophase in meiosis is the crossing over. So this is important. This is happening here and happening here. And it looks like I forgot one right here because I'm a mess. Boom, 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 boom. There it is for reals. Okay. And then we got metaphase. Remember meta means middle. So they line up in the middle of the cell. Okay, then we've got anaphase. Anaphase means away, so they're being pulled apart. And then telophase, cytokinesis, is when they're going to split into two. Now, an important thing to remember here is that up over here in metaphase, if we would have switcherooed these guys around, so we would have had a blue and red, then over here it would be blue over here and red over here. So order doesn't matter as far as the telophase. It's just a matter of whatever happens in metaphase. So whatever happens in metaphase is going to determine how the cells split. Now remember, we've got to do this 2 times for meiosis. Okay, so then we've got prophase and metaphase 2. Prophase again is we're going to be making our chromosomes. Metaphase is going to be lining up in the middle. Anaphase is apart or away, so they're going to be split, just like in mitosis. So this should be kind of familiar to you guys. And then in telophase 2 and cytokinesis, we've made our gametes. So we have 4 possible gamete cells. Remember, for men, this is what creates your sperm cells. And for women, this is what creates your egg cells. So we have 4 sperm possibilities, or 4 egg possibilities. And if you notice, they all look different. So we have all reds here. We have red with blue tips. We have blue with red tips, and we have all blue. So each one of these cells is going to make a different looking baby, or a baby with different features. And that's one of the cool things about meiosis. All right, so if we're looking here, we know that egg and sperm cells, they're gametes because they have half the number of chromosomes that the full organism has. Okay, so if you're human, you have 46 chromosomes. So that means that your egg cells have 23 and your sperm cells have 23. All right, crossing over. During prophase 1 of meiosis, crossing over occurs. And then what is crossing over? Hey, it's in this picture. So chromosome segments from homologous chromosomes. Okay, homologous are going to be 2 chromosomes together. Their homologous means the same size and shape from each parent are swapped. Okay, so in this case, what they did is they took these 2, the orange and the yellow, and they crossed them over. Okay, and then how did crossing over change the chromosome? So we have new gene combinations are made by mixing mom's and dad's genes to make you unique. Other ways to get variety in the cell, crossing over makes cells different, like we saw in our gametes. This calls we call this creating variety. Okay, there are other ways meiosis produce variety. One way is the law of independent assortment. This means that chromosomes can line up in any order in the middle of the cell if the homologous partners or line up together. Okay, so in this case, let's say we've got yellow from mom and blue from dad. So we've got the lining up here. We've got two big guys over here and two little guys. Or we've got, oh, I see, or down the middle. So it depends upon how it splits. So this guy, it's split here. And this guy, it's split here. But what you notice is that these two were swapped. So here we have yellow, blue, and here we have blue, yellow. Okay, and that's going to create some diversity in genetics. Okay, so, and then it's asking us, are there other ways you could have arranged your model chromosomes? Now, we couldn't really, since we were only using 2 chromosomes, but in real life with 23 chromosomes, there are a lot of options. So basically, if we roll the dice 23 times, that's how many options we've got there. So why is it beneficial? It creates genetic diversity, which is essential for evolution and natural selection, because we need to have variation. If we don't have any variation, then one disease or one situation can wipe us all out, and we'll get back to that later. Explain why meiosis is necessary in creating sex cells. So this is because they only need half the chromosomes to pass on to the baby, which is why the cells divide twice. So because if I had 46 from mom and I had 46 from dad, then as a baby I would have 92 chromosomes. That would be a completely different organism. So in order to do that, in order to make the 46 chromosomes that I need to be a person, I just need 23 from mom and 23 from dad. Okay? All right, next we're going to do a little comparison here. All right? So we know they're different because in mitosis, remember we only split 1 time. In meiosis, we split 2 times. Okay, so what process is this? The first one is mitosis. The second one is meiosis. This makes body cells. Mitosis makes Henrietta's body cells, like her cervix, where her cancer was. And meiosis makes Henrietta's egg cells, which will go on to become her children. Okay, then I just added some notes here. I got PMAT, so prophase, metaphase, anaphase, telophase, prophase, metaphase, anaphase, telophase times 2. Just so we can remember some differences here. So we labeled them, yada yada. Where did Henrietta's mutation happen? So in order to get cancer, her tumor cells were in her cervix, which is part of her body, not part of her ovaries. Then it says label where you think Henrietta's mutation happened in the diagram. Okay, so over here in this orange star area, this is where her mutation happened in prophase when DNA replicates. Okay, so what happened was there was a copy error. Okay, so remember when we were copying DNA, so we have one side of DNA and then it opens it up and then we make another copy. Okay, so if there is a mutation, then that could be causing cancer in our DNA replication. So that's where it's going to happen. So in prophase or in interphase, depending upon where the cell is in its cycle. Okay, so today's goal is to show why Henrietta's children do not have immortal cells like she does. Okay, all right, so something that I realized is I should have put the word steps here because that would have made way more sense. But so if you did something else for yours, then that's fine. This is just what I did. There's another Amoeba Sisters video for you guys because I like how it does it side by side to show you mitosis and meiosis. Okay, so prophase, metaphase, anaphase, telophase, cytokinesis. Now, something I could write for every single difference. So the differences are here in this bottom row. Every time I could write in meiosis, it happens twice. Okay, so prophase happens twice, metaphase happens twice, anaphase happens twice, telophase happens twice, everything happens twice. Okay, but a couple of big things. Okay, this is what's happening in each of the steps. Boom, 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 boom. So this is what's happening. And then a couple of key differences. We've got the crossing over. Get a highlighter here. We've got the crossing over. That's a big difference. We've got 2 by 2, like Noah. And then then we have single file, which is very much like mitosis. Then in anaphase, in meiosis 1, we pull apart homologs. And then in meiosis 2, we pull apart chromatids. And again, in the second step, it's just like mitosis. In telophase, 1, they both have chromosomes in them. And then in telophase 2, it's just like mitosis. Okay, and then the last but not least, in mitosis, after cytokinesis, we have 2 new cells with 46 chromosomes each, and then in meiosis, we have 4 new cells with 23 chromosomes each. All right, then application. Why do we need mitosis? In order to create body cells. Why do we need meiosis? To make sex cells, to make babies. Mitosis we are going to use to heal wounds and replace dead and damaged cells. Meiosis is needed to make babies. The reason that all of these are even numbers, half from mom, half from dad, it has to be even. If it's not even, if it's an odd number, then there's usually some sort of chromosomal mutation. Like, for example, in Down syndrome, they have an extra chromosome, and so they have 47. Okay. Why is the reduction in chromosome number important? It's kind of like I said before, we don't want to have 92. Why is crossing over important? Why do we care about genetic diversity? We care about it because bananas, and you'll see what that means later. When you identified a mutation, did it show up in every cell? Not at first. Okay, so we have the original DNA here in orange. Then we have the replication. Then we have 2 new cells. Then if I take this cell and I replicate it again, then I'm going to have a fully mutant body cell, and every cell that comes after this one is going to be mutated. This is going to make a tumor. Okay. And then the mutation would have had to be in her ovaries if she would have passed it on to her kids. And so no, her cancer cannot be inherited. Okay. Then we've got some vocabulary again for our wrap up and review. We need to be able to create sex cells. You can tell me a bunch of differences. For our review questions, we've got one about attached earlobes. And this is kind of a Punnett square review. So unattached earlobes are dominant, so they're capital E. And attached earlobes are recessive, so they're lowercase e. And remember, heterozygous, that is one big E, one little e. So it's wanting to know what are the chances of the child having attached earlobes. So that's 1 square here, 25% chance. All right, then, why is it important to have genetic diversity in a population? This is a huge question for understanding evolution and ecology and things like that. So I put this cool video here called The Terrifying Truth About Bananas to help you understand why genetic diversity is important. So take a listen, see what you come up with, and I'll see you next time.